Tasmanian tiger, Atlas bear, stellar sea cow, and the ground sloth. All these beautiful species are no longer with us. They're extinct, gone from our lovely planet forever. If there was a way, though, would you bring them back to life?
I think messing with nature on that level could get a little hairy. I'm very much one of those people who is like, if it's gone, it's supposed to be gone.
If we can support them and take care for them and not just bring them back for our own benefit.
You've probably heard of Steven Spielberg's Jurassic Park, where a team of crazy scientists clone a bunch of dinosaurs and let them run wild on a fictional island in the Pacific.
Welcome to Jurassic Park.
Nobody move a muscle.
That scene with the raptors where they were hunting the kids in the kitchen, that was absolutely terrifying. So I think we're all in agreement that we do not want a Jurassic Park situation on our hands.
It just seems like a recipe for disaster.
If they were to come back, humans would be extinct.
I think they'll outsmart us eventually. Reclaim the Earth.
No, thank you. So, excluding dinosaurs, what other extinct animals would you like to see again?
The megalodon.
I would bring back the woolly mammoth because it could provide enough fur and meat, especially in the cool environments.
I'm going to go with a woolly mammoth.
The dodo.
Bringing back the dodo could help us make up for our mistakes. The bird went extinct as a direct consequence of human actions. Is it possible to use science and ancient DNA to heal the damage we've caused? From the Sowers Institute for Medical Research and KCUR studios in Kansas City, this is Seeking a Scientist, a podcast where science fiction meets reality. I'm Dr. Kate Bieberdorf, aka Kate the Chemist, and in this episode, we're investigating de-extinction, which I'll admit is a new vocab word for me. It's the science of resurrecting extinct species, and more impressively, the science of inventing new types of previously extinct species. This mostly theoretical idea has been around for over 40 years, but it finally earned a name when the TEDx De-Extinction Conference went viral in 2013.
Biotech company Colossal says it will have the science to de-extinct woolly mammoths by 2027.
Woolly mammoths have been extinct for about 10,000 years.
But now, one extinct animal might be getting a reboot.
After so much speculation, what exactly is possible? Today's expert, Professor Beth Shapiro, has been working in this field for years, but she has mixed feelings about the term.
So this idea has been there from the beginning and now has a name, de-extinction. I don't really like it. The reason I don't really like it is because it's impossible to conjugate. How would you say that you achieved it, right? Did you say you de-extincted something or de-extinctified it? I don't know. None of those things are things that roll smoothly and quickly off the tongue.
That's fair. If Beth doesn't like it, then I don't like it. See, Beth's a MacArthur Fellow, which scientists commonly refer to as the Genius Award. She's a Howard Hughes Medical Institute professor and recently became the Chief Science Officer for Colossal Biosciences. Basically, she wears a lot of hats. But if you ask her what her title is...
Most often I use the word paleogeneticist because it's confusing. People don't know what it means. But if you think about it and break it down, it makes sense. Paleo is old stuff. Genetics is I study DNA.
When she says she studies the DNA from old stuff, she's talking about the mammoth bones that she digs out of mining zones in Siberia. We'll get into how she actually extracts the DNA from skeletons. But first, let's talk extinction. Scientists estimate that over 5 billion plants and animals have vanished. That's 99.9% of all species that have ever existed on Earth. Unfortunately, and sadly, that's just part of life. We've had 5 major mass extinctions, but you're probably most familiar with the Cretaceous-Paleogene extinction event, the one responsible for killing all the dinosaurs. That was nothing compared to the Great Dying, the nickname for the Permian-Triassic extinction, where 96% of all marine species were lost and 75% of land species died too. In the End-Triassic extinction, we lost all of the conodonts, which I've heard described as fanged nightmare eels, so I'm not going to lose any sleep over their extinction. But in the last century alone, over 500 vertebrate species have died. We lost the golden toad when its climate became too dry, the giant Pinta Island tortoises when humans introduced goats to the Galapagos Islands, and the Caribbean monk seal when hunters started targeting them for their blubber. I don't know about you, but it really bothers me that I'm never going to see any of these species with my own eyes. Because unlike the dinosaurs that were taken out by an asteroid, the golden toad, the Pinta Island tortoises, and the Caribbean monk seal were all catastrophically affected by the introduction of humans. Whether we've overhunted them, destroyed their habitats, introduced predators or diseases, humans are at fault for why a lot of adorable species are no longer here with us.
Did you end up with a favorite? Is there one that you would like to bring back?
Me? Definitely the moa. So we just learned about them when we were in New Zealand. So 12 feet tall, never developed wings so they couldn't fly. Amazing, right? Okay, what about you? What's your favorite?
Oh, it's tough for me to pick. What would be the coolest? I love the dodo. It's the very first species that I ever worked on as an ancient DNA scientist. I got a tiny bit of DNA out of the dodo that is in the collection at the Oxford University Museum of Natural History and was able to show that the dodo is a pigeon. So at the same time, really interesting and also maybe a little bit disappointing, right? It's a beautiful, really interesting bird, but it's a pigeon, right? I'm just kidding. Pigeons are amazing. Also, it's the icon of how awful we can be. They went extinct within just a few decades of people first appearing on Mauritius, which is the only place that dodos ever lived. A few decades, right? And it's remembered as this very sad, stupid bird. It probably wasn't stupid. It just didn't know to be afraid of us because it never had any predators before. And I just feel terrible for it. I think if we were to bring that back, we could make it an icon of the power that our technologies and determination could have to reverse some of these atrocities that our species has committed on the planet since we've existed.
So it's almost the symbolism that you like of it. It's not necessarily the bird. Is that what I'm hearing?
The symbolism,
okay,
but also the bird. I mean, come on, you've seen the pictures of this bird. Wouldn't you like to have a bird that's kind of a little bit higher than your knee that sort of wanders around with you, looking at you with its big, cute face and floofy tail? And
I can see it. I can definitely see it. By now, you're probably wondering, how exactly is it scientifically possible to make this happen? Well, Beth and her colleagues have to weigh a lot of factors when considering bringing a species back. Like, what would happen to New Zealand if we reintroduced the moa to the South Island? What would the bird eat? Would it have any predators? Would it be vulnerable to modern diseases?
For example, do we know why it went extinct in the first place? This is important because we wouldn't want to spend all of this time and energy and effort bringing this thing back if it was just going to become extinct again. For the dodo, for example, we know that dodos became extinct not because people ate them, but because when we arrived on Mauritius, we brought with us things like rats and cats and pigs and monkeys and dogs, and dodos couldn't fly. They laid a single egg in a nest on the ground, and that was an easy meal for all of the stuff that we brought with us.
Okay, so let's say we decide to bring back the dodo. Step number 2 is figuring out where we're going to put them. They used to live on Mauritius, a small island off the coast of Africa near Madagascar.
So if we're going to bring a dodo back, we have to be able to find a place where it can live that doesn't have these other invasive species that would just eat its eggs. So it doesn't make sense to go through this project unless you've done this. About a month ago, I was in Mauritius working with people from the Mauritian Wildlife Foundation to visit some of the sites where they're doing incredible conservation work today. It's a really amazing place. They've reintroduced giant tortoises, not the species that went extinct, but related species from another island. The plants that evolved alongside tortoises are doing better. And some of the invasive plant species that have been introduced that don't have anti-tortoise mechanisms are not doing so well now that tortoises are there to eat them. So putting this species back in this habitat has really helped the whole ecosystem to become revitalized. And I think this is a compelling reason to think about reintroductions of extinct species or species that have some of these extinct traits.
So in this process, it's crucial to look at the species within its environment. Does this habitat still exist? Would the animal be able to survive if it was introduced today? Will we have dodos in the next 500 years?
Absolutely. Way sooner than 500 years. I mean, as long as you're willing to accept a Nicobar pigeon that has been modified to have some dodo phenotypes, we are 100% going to have a dodo by then.
Whoa.
I mean, the goal of all of these projects, I think, should not be to try to recreate some ecosystem fixed for the past. The past is over. We need to think about how do we use these technologies to create ecosystems that are resilient and robust for today and for wherever the world is going.
That's asking, what ecological purpose does the extinct animal have in that specific ecosystem? Who, or maybe what species, will benefit from the re-addition of the dodo bird? One way to study the idea is to create a park and start putting animals in it.
Welcome to Jurassic Park.
No, not that kind of park. I'm talking about Pleistocene Park in northern Russia.
Pleistocene Park is run by a pair of scientists, Sergei Zimov and his son Nikita, who have been working to try to re-engineer these ecosystems. Their hypothesis is that if we put these large herbivores and potentially carnivores into this ecosystem, that their existence will cause the regeneration and resurgence of the tundra, the steppe tundra ecosystem that used to be there. So they put bison and Arctic adapted horses and a couple different species of deer, and are really hoping someday for mammoths and woolly rhinos and other species to exist in some form so that they can have them in their park, because they see these larger herbivores as really keystone to the species because they were bigger and they were knocking things down and really churning the soil more and distributing nutrients and distributing seeds, and they would like to have these species back.
It's important to realize that we're not just talking about bringing animals back from extinction. The idea is to use de-extinction to kickstart an entire ecosystem. At the moment, all 10 types of animals in the nature preserve are regular old herbivores that we have simply relocated to the park. They have fur goats, plains bison, moose, musk oxen, sheep, Bactrian camels, yaks, reindeer, Yakutian horses, and Kalmykian cows. Both Sergei and Nikita are working effortlessly to make sure that when the next dinosaur, dodo, or mammoth is born, we're ready for it.
His project is ambitious, to recreate an ecosystem that disappeared 10,000 years ago with the end of the Ice Age.
We are not trying to invent a bike. My father and I want to restore ecosystems which were on this place before first humans came to this land.
Beth says de-extinction is actually a word the media generated back in the 1980s when Berkeley professor Alan Wilson found some DNA from a quagga. Since this half-horse, half-zebra had been extinct for more than 100 years, Alan's discovery was a huge deal.
This was the first time that anybody had actually gotten DNA from something that was dead. And so it opened up this whole new realm of potential research. Can we get DNA from stuff that's been gone for a long time? In fact, this work inspired Michael Crichton to write Jurassic Park.
In this iconic film, scientists find dino DNA in a mosquito that's been perfectly preserved in amber. This is how they broke it down in the movie.
100 million years ago, there were mosquitoes, just like today. And just like today, they fed on the blood of animals, even dinosaurs. Bingo. Dino DNA. We used the complete DNA of a frog to fill in the holes and complete the code. And now, we can make a baby dinosaur.
Then with a little Hollywood magic, Jeff Goldblum had quite the experience in a park full of hungry dinosaurs.
Come on. We gotta get out of here. Now, right now.
Beth is often quoted as saying that her goal is to explain de-extinction in a way that separates science from science fiction. Basically, she wants to differentiate the scenes in Jurassic Park that are definitely fiction from the parts that represent a real area of research. It's a tall task, though. Steven Spielberg essentially introduced the world to Michael Crichton's idea of de-extinction, but it was massively flawed.
In fiction world, in Jurassic Park, they had little tiny sequences of dinosaur DNA. No, they didn't. And then they filled in the gaps with frog DNA, which was a weird choice because even at the time we knew that birds were dinosaurs, but whatever, dinosaur DNA and frog DNA, and we just filled in the bits and then it made perfect dinosaurs.
The truth is, we cannot bring dinosaurs back. Full stop. In the next 100 years, is there any chance that we will have a park that is anything like Jurassic Park?
With animals from the Jurassic?
Your call.
I mean, there already is Pleistocene Park, but will we ever have dinosaurs? Like real dinosaurs with dinosaur DNA? No, because there is no DNA in dinosaur remains. Dinosaur remains are rocks, and rocks don't have DNA. I'm sorry.
In order to do this with modern technology, we would need a living dinosaur cell. And since dinosaurs went extinct 66 million years ago, that's not going to happen. So let's break down why. As soon as the species dies, its DNA begins to degrade. If the animal passes away in the tropics, the DNA will decay quickly because sunlight is DNA's worst enemy. Animals that die in the freezing cold, like Siberia, have a better chance of being brought back to life because their DNA decays much more slowly. But there's time constraints too. Scientists estimate we're only able to sequence DNA for species that lived within the last 6.8 million years. After that, the DNA is going to be too old, too damaged to be useful. Therefore, all dinosaur DNA is long gone. But what about the species we lost more recently? Can we bring some carefully selected extinct species back to life?
Well, probably not. Not in a way that means that this species is going to be 100% identical in every way to a species that used to live. But we can, using the tools of modern genome engineering and cloning and other really cool stuff that has been developed over the last decade, we can probably de-extinct or de-extinctify or whatever you want to say, extinct traits. We could take a thing that's alive today and gradually tweak its DNA so that it looks and acts more like something that is now extinct.
This part is so cool. Hypothetically, let's say giraffes were starving because they had eaten all the food they could reach. One solution would be to alter the giraffe DNA by inserting tiny pieces of the dinosaur Brontosaurus's DNA so that the giraffes could grow even longer necks. But unfortunately, this specific scenario will never happen because our current technology cannot handle how damaged the dino DNA is. And we need the DNA because
this is what provides the cells the instructions that they need to make the proteins that make us look and act the way we do.
These instructions are encoded by 4 molecules: adenine, A, cytosine, C, guanine, G, and thymine, T.
Back to life, back to reality.
And these letters, the order of them and the number of them changes slowly over time. In us, these letters are arranged into our chromosomes, right? And all together, our chromosomes, which live inside the nucleus of our cells, make up what we call our genome.
Your genome is like the instruction manual for your body. The chapters of the manual are the chromosomes. Chromosomes are made up of genes, and each gene has a specific task, like determining your eye color or diabetes status. You can think of the genes as the sentences on the pages within the chromosome chapters, and the letters that make up the sentences are the DNA, a teeny tiny 4-lettered alphabet, A, C, G, and T. So the genome is your manual, the chromosomes are your chapters, the sentences are your genes, and the letters are your DNA. If we say ancient DNA, we're talking about DNA that has been extracted from a really old specimen. It's something that scientists stumble upon, usually in the Arctic. It's not intentionally preserved for later use. So, if we're really trying to pick the perfect species to bring back to Earth, it's not going to be one of the dinosaurs or the dodo. It's the frickin' mammoth.
We get these tiny little fragments of DNA, and we use a computer, and we line all these tiny little fragments of DNA up against the genome of the closest living relative. So we have a map. That's the closest living relative. Think of it as like the picture on the front of the puzzle box. That's their closest living relative genome. It's an Asian elephant if it's a mammoth, right? We have a picture of an Asian elephant, and we have a trillion, gazillion, billion tiny little pieces of elephant DNA that are all mixed up with 10 trillion, gillion, bazillion pieces of microbial DNA that we also extract from this mammoth DNA. So we've got a bazillion, gillion, gogol piece puzzle with the wrong picture on the top, right? But we can still do this exercise with a computer because computers are smarter than us. And we line up all these pieces and we get what approximates a mammoth genome. And then we can line that up against an elephant genome. We can ask where they're different from each other. But then when we start the editing, we don't start with the fragments of mammoth DNA. We start with a living cell. We start with an Asian elephant cell growing in a dish in a lab. And we use the tools of genome engineering to gradually cut and paste our way from that elephant cell's genome to a genome that more closely resembles a mammoth. And we do that by using all the instructions that AI has now given us about what it is that we need to edit in that genome to make it express those mammoth traits. So then we have a living cell in a dish in a lab that is mostly Asian elephant and a little bit mammoth-like.
In Beth's best-selling book, How to Clone a Mammoth, she discusses how in 2021, Anders Gutterstrom and Love Dellin from the Center for Paleogenetics released the genomic data for 3 mammoths, 2 of which were more than 1 million years old. What's particularly neat about this is that they discovered protein coding changes that woolly mammoths relied on to survive in the horrendously cold temperatures. With this information, scientists can now handpick a few of the mammoth's most favorable traits that can help its closest living relative, our buddy, the Asian elephant. So, if due to climate change, the Asian elephants start to migrate north, maybe we could help them with the transition by implanting some mammoth DNA that helps them weather the intense cold. Imagine an Asian elephant covered with luxurious mammoth hair, and you've got the idea. To get this phenotype, which is a specific set of observable physical traits like long mammoth hair, one would think you have to head to Siberia to find the mammoth bone that contains this information.
It's not necessarily true, though, that we need that genome sequence to come up with that phenotype, because we could engineer that phenotype by understanding genome sequences from other things. This is one of the hardest problems in biology today, is trying to map a DNA sequence to a trait, a behavior, or a phenotype, the way something looks or acts. And we can use these large computer scans to figure out if we can map DNA sequence changes to the physical changes or the changes in disease that we see in individuals. And then that allows us to have hypotheses about what genes might be associated with what traits in other species. So we can use computers, maybe even AI at this point, to try to help us take in all of this huge amount of information and use that to narrow down the list of edits that we have to make to a genome sequence in order to turn it into something else.
In other words, Beth needs to go to Siberia, find mammoth bones, extract the DNA, use a computer to analyze the DNA, and then go line by line to determine which set of molecules produces the dense, wavy mammoth hair before she can even think about sneaking them into an Asian elephant genome.
If we want to be faithful to evolution of the past, then maybe we want to use that exact genome sequence.
Mammoth 2.0 won't be exactly like the 13,000-pound species of yore, but it has potential to be a super close cousin. Coming up, how to clone a mammoth. I cannot stress enough how much work Beth puts in to try to clone these mammoths. Like, she has to go all over the world trying to get this DNA. She's dealing with mosquitoes.
We go up in the summer where it's not freezing, but there are about 7 million mosquitoes per cubic centimeter. So, pick your poison or stabbiness in this particular.
And curious men with large guns.
There was this brief moment of, oh my God, we might die. And that quickly changed to, way, we know each other, we're great friends.
On these trips, Beth works closely with the local miners to search for well-preserved mammoth bones, not tusks. The best ancient DNA is found within the bones.
Most of people who work in ancient DNA and me spend a lot of time in the Arctic, in places like Siberia, Alaska, Canada's Yukon, trying to find recently exposed bones from these animals that used to be alive. And these bones look great. You go up and we can collect bison bones, horse bones, mammoth bones that look like the animal died and was defleshed by some carnivore just yesterday. And we know that these are going to have really well-preserved DNA.
The silver lining of climate change is that the increase in temperature is causing the permafrost to melt and exposing really well-preserved animal skeletons. Beth and her colleagues take great care to avoid contamination when collecting the samples. After all, her entire research depends on getting those bones back to the lab so they can begin searching for DNA. They start by extracting a small sample from a compact part of the bone. Then they smash it to smithereens. The goal is to achieve the most surface area possible. After a chemical workup, they extract the DNA and then use a technique called PCR, polymerase chain reaction. This tool makes scientists' lives so much easier because it basically takes the DNA and goes Control-C, Control-V until they have large amounts of the DNA to study. And this is where the fun really begins.
And then we use cloning, the somatic cell nuclear transfer, the same thing that brought us Dolly the sheep, to transform that living cell into an animal.
She's an identical copy of an older sheep.
Dolly is the name of the first mammal, yes, a sheep, ever to be born as the result of cloning.
Cloning is this process of somatic cell nuclear transfer where you take an egg cell and you remove the nucleus that has the DNA and you inject a different nucleus and then you inspire that cell in some way to reset itself and become the type of cell that can start to divide and differentiate and become a whole animal.
For Dolly's case, sheep A donated the nucleus and sheep B donated the egg. The nucleus and the egg were then fused together and that eventually formed the embryo. The embryo was then implanted into surrogate mama sheep C. 5 months later, when Dolly was born, scientists were able to confirm that she was the identical clone of sheep A, the nucleus donor. They shared the same DNA.
So cloning is part of the process, and you will have to clone in order to get a mammoth, but you're cloning an edited Asian elephant cell. So what do you end up with in the end? It's an Asian elephant that has mammoth DNA. Is it a mammoth? Is it mammoth 2.0? Is it an Arctic adapted elephant? I think, yeah, it's all of those things.
So are mammoths and Asian elephants similar sizes? Would there be any concern about using a mama Asian elephant and then trying to build a mammoth using her body?
This is a great question. So I called Adrian Lister at the Museum of Natural History in London, and who's the world expert on mammoths, and said, Adrian, tell me about the size differences between these animals. I'm really intrigued, and I, of course, want to know this isn't going to work. And Adrian's quiet for a moment because he's that kind of person. And he goes, no, they're about the same size. That'd be fine.
But not all species that went extinct could be safely cloned in birth today. I'm talking about animals like the Steller sea cow, which was absolutely massive. This species used to fill our oceans and unfortunately went extinct a couple hundred years ago.
Steller sea cow was large enough that it would feed a crew of 30 men for 30 days, like really giant thing. Closest living relative is a dugong. That a baby Steller sea cow growing up inside a dugong maternal host would be about the same size as or slightly larger than its mom at birth.
The Steller sea cow might be off the table, but Beth thinks that the Asian elephant mamas could handle a mammoth 2.0 embryo, though we're still quite a few years away from that becoming a reality. Meanwhile, on a much smaller scale, we're currently using this idea to make ecosystems more resilient by bringing more genetic diversity. One of the most successful programs is Revive and Restore's Black-Footed Ferret Project.
This is a little carnivore that used to live across the American plains that everybody thought was extinct. And they discovered another population that was still alive near Meeteetse, Wyoming. But they're still in trouble. They're still in trouble because there's only 7 founders, right? And that means that breeding with close relatives is inevitable. And the second problem is that people introduced plague into their habitat. And so when these individuals are released into their habitat, they get plague and then they die. And what's cool about this is there are black-footed ferrets from the original project that are frozen in tissue culture at the San Diego Frozen Zoo, and these can be used to clone and resurrect extinct diversity that can be injected into the captive breeding population. And the domestic ferret, which is an evolutionary cousin of the black-footed ferret, is naturally immune to plague because it evolved alongside plague and evolved a way of escaping dying from plague.
The idea is to introduce a combination of the plague immunity trait from the domestic ferret and a few diverse traits from the frozen black-footed ferrets to create a resilient black-footed ferret that can survive in today's habitat. They successfully cloned 3 ferrets and are hopeful that they are on the right path for generating more genetic biodiversity. With all this in mind, we can't pretend that this science goes without controversy. Speeding up evolution, bringing back something that no longer exists, and editing genetics will all have significant effects on the ecosystems we're in.
Our scientists have done things which nobody's ever done before.
Yeah, but your scientists were so preoccupied with whether or not they could that they didn't stop to think if they should.
People who are critical mostly say things like, you're never going to create something that is 100% identical to a species that exists. And my response is, I agree with you. And then people say, all of the money that's going into de-extinction, and I guess they're saying all of this money should go to traditional approaches to conservation. And I say, you're right. That much money should go to traditional approaches to conservation. We have to think hard about what we're doing before we do it.
Maybe you are listening right now having your own doubts, but I challenge you. Think about the big picture. Our planet's creatures are in trouble, and maybe the solution is hiding in the permafrost of Siberia.
Today, if you look around us, we see habitats around the world that are in trouble, and we don't really know where to begin, right? But the past is a time period that also experienced really rapid, tumultuous perturbations to habitats and climates around the world. And we can use that past as a kind of completed evolutionary experiment. If we get DNA from loads of bison and horses and mammoths that lived during the period of rapid climate warming out of the last ice age and into the Holocene, and we ask questions about why some of these species survived and others didn't, what makes communities more robust and resilient in the face of these rapid and large-scale perturbations to their habitat, then we can use that information to make more scientifically informed decisions about how to protect and preserve ecosystems and species in the present day. And I think this is a really important contribution that you can get by studying the past.
Chinese philosopher Confucius is famously quoted for saying, "Study the past if you would define the future." We all know that history repeats itself, but we don't have to keep making the same mistakes. Let's say, for whatever reason, you're able to come back to planet Earth 500 years from now. Where do you want your field to be? Like, what would be the best case scenario?
I would love for us to have figured out that map between genotype and phenotype, because that is the map that's going to tell us how we can use our technologies to do things like stop genetic diseases, create species that are robust to chemicals that have been introduced into their ecosystems, to pathogens that people have introduced into their ecosystems, or even use phenotypes from the past to help things that evolve to be able to be fit and robust in the habitats of the future.
It took us 32 years to sequence the first human genome. But thanks to next-generation sequencing, we can sequence a person's genome in a single day now. We are at the forefront of the field of paleogenetics, and I really hope Beth accomplishes her dream of determining which gene is connected to which mammoth physical trait. So eventually, a version of the woolly mammoth can once again roam our planet. Okay, friends, there's one more thing I haven't touched on yet. I know the topic of cloning and messing with DNA can bring up some conflicting feelings in people, and I want to acknowledge that and validate your thoughts. But here are my 2 cents. I am so jealous that Beth gets to go on these amazing trips and work with international colleagues and literally go hunting for mammoth bones. She's basically Laura Dern's character from Jurassic Park. Her work in the lab, extracting ancient DNA, sequencing the genome, and then attempting to match physical traits to genetic code is HARD, hard. And whatever your reservations, what we could learn from her results could literally save critically endangered animals like the Sumatran elephant and Asian elephant. So for me, it's hard not to be on board with these areas of research. Plus, Beth was just really fun to interview. What percentage of Jurassic Park is accurate or possible?
0%.
Do you watch true crime movies and shows?
Love true crime. The one main thing that you hear about in true crime is that you can't get DNA out of hair unless it has a root. Not true. There's loads of DNA in hair. It just is really tiny, short fragments. And so the way that, until recently, people working in the crime industry have been characterizing DNA hasn't been possible from DNA preserved in hair. But this discovery, this work is poised to absolutely change the way that we do crime scene investigation. You are not going to be able to get away with murder.
You heard it here first. Okay, so now I have a couple like this or that, and they're all extinct animals. So I just want you to pick your favorite.
Pick my favorite extinct animal. This is like asking you to choose between your cat and your children. Okay, go.
Let's see if you can. You can pass on anyone you don't want to. Okay. Giant bear or saber-toothed cat?
Oh, I love them both. I have DNA from giant bears and no DNA from saber-toothed cats yet, but saber-toothed cats are so cool. So, saber-tooth.
Okay. I love that. Moa or golden toad?
That's a hard one. Golden toad. Moa. Okay, why'd you make me choose between these things? I'm gonna... where did the golden toad live?
I don't know. I just thought it looked cool, so I thought you might like it. Let me give you one more. The quagga or the elephant bird?
Elephant bird.
Why?
Quagga is just a type of zebra. Quagga is lovely, whatever, but we have other zebras. Elephant bird, have you seen one of those eggs? Amazing. Amazing.
Listener, I had not seen an elephant bird egg. They are shockingly large. I'm going to post a picture of them on our Seeking a Scientist Instagram page because you've got to see them with your own eyes. One egg can hold 2 gallons of liquid. And the golden toad, it's from Costa Rica, which is creepily close to Jurassic Park's fictional location. But you don't have to worry about that amusement park becoming a reality anytime soon. Now, thanks to Beth, we can all sleep safely at night knowing that scientists are only dreaming about creating the next T-Rex. However, I cannot say the same thing about Steven Spielberg.
It would be wrong to go back and bring the dinosaurs back in real life, but not in a movie.
Welcome to Jurassic Park.